Okay, so the first part of section 3 is going to talk about viral replication. So I'm going to divide section 3 up into two different videos, part A and part B, just because it's a little bit longer. There's two different types of viral replication. There's the lytic replication and then there's lysogenic. So we'll start off by talking about the lytic. But in either case, anytime we're talking about viral replication, we're just talking about how the virus turns the host cell into a virus-producing factory. So there are different methods that viruses can use depending on what type of nucleic acid that they house, but most of them are going to follow a similar kind of game plan. So that's going to start with attachment, then entry, then synthesis, assembly, and then release. So we need to always begin with the virus finding a specific receptor on a host cell, and that's that lock and key mechanism I talked about previously. And this attachment to a cell is very, very specific. If the receptor doesn't fit, then it will not be infected at all. Once it's attached, the virus or its genetic material is going to enter into the cell. So either by injection, if we're talking about a bacteriophage, or other mechanisms when we talk about animal viruses later on. And then after the virus enters into the host shell, it's going to use the host shell machinery in order to replicate its genome and produce viral proteins. And then all of these components that it just made using the host machinery are going to kind of self-assemble to form new virions. And then finally, the virions are going to be released, usually through cell lysis, if we're talking about the lytic stage, which is going to cause the cell to kind of burst open. The other option is through budding. So that's how enveloped viruses will exit a host cell. So talking a little bit more about the lytic cycle, this cycle is really fast and it's extremely destructive, as you can imagine, because it's going to cause cell lysis. So it's going to begin in this example with the bacteriophage attaching to a bacterial cell wall by using its tail fibers. Once it finds the right receptor, the phage is going to inject its nucleic acid, in this case I believe it's DNA, into the cell, and it's going to leave behind its empty capsid outside of the bacterial cell. Then the viral genome is going to really quickly shut down the bacterium's normal processes and kind of redirect the host enzymes to make its viral components. Then the new phage particles are going to be assembled and the cell is eventually going to lyse open, releasing hundreds of offspring that will go on and infect new cells. Okay, so the lytic cycle, it can be visualized as a viral growth curve. So that's what you're seeing on this slide here. What it's showing you is the time down here on the x-axis and then the number of infective virions on the y-axis. It is in a logarithmic scale. So you'll notice what we're starting with is only 1. So the virus is going to infect a host cell and inject its nucleic acid, or the whole virion is going to come in. So once it enters into its host cell, then we're going to have 0 as far as how many viral particles we have available, because what they're counting is the number of viruses that are outside of the cell itself. Then we still continue to have 0 cells outside of the cell, or 0 virions outside of the cell during the synthesis and assembly phase. This is usually referred to as the eclipse period, and no new viruses are going to be detected outside of the cell, which is why it's at 0, because the virus is still replicating in its genome and producing proteins inside. The next step is going to come, what's usually referred to as the rise period. So when the host cell lyses, it's going to release a flood of new virions. And then eventually it's going to plateau right here once most cells are infected and destroyed. So this pattern is really important because it explains how infections can escalate so quickly. The viral population can go from being completely undetectable to explosive in a short period of time. Okay, so not all phages are in a rush to kind of destroy their host cell. Some are going to undergo the lysogenic cycle in place of the lytic cycle, and it's usually a little bit quieter at first. So after attachment entry, the phage DNA is going to integrate into the bacterial chromosome, And once we have the phage DNA in the bacterial chromosome, we then refer to it as a prophage. And so instead of taking over the cell, it's going to lie dormant while the bacterial cell continues to divide. And each daughter cell is going to end up inheriting the prophage, which is going to allow the virus to quietly kind of spread without killing its host. And this can go on for several generations. But if something like UV light, radiation, or stress occurs, the prophage can be triggered to reactivate. And when it does reactivate, it's going to switch to the lytic cycle that we just talked about and then burst or lyse the host cell. So this slide kind of breaks down the lysogenic cycle down visually. So again, first the virus is going to attach, inject its DNA. Instead of taking control of the host machinery, the viral genome is actually going to integrate into the host chromosome and therefore become a prophage. Then the bacteria is going to divide. Both daughter cells carry the viral DNA. If an environmental signal is detected, like something like DNA damage, the prophage is going to be induced to leave the chromosome and then shift into that lytic cycle. So lysogeny isn't just about hiding. It can actually change the behavior and the traits of the host bacteria. So when the phage DNA integrates into the host genome, it's going to sometimes bring genes that alter the bacterial phenotype. And this can turn a harmless bacterium into a pathogen by adding toxin or virulence genes. And this process is called lysogenic conversion. When the prophage is later induced by some sort of stress like UV light or X-rays or even mutagens or some sort of chemicals, it's going to cause it to leave the chromosome and enter the lytic cycle, destroying the host while spreading both its viral particles and any new traits that it's now carrying. Okay, so when it comes to animal viruses, the overall process of replication follows the same general 5 steps as in bacteriophages that we just talked about. So attachment, entry, synthesis, assembly, and release. But since animal cells lack cell walls and many animal viruses are enveloped, the strategies that they use are a little bit different. So instead of injecting genetic material through a wall like phages do, animal viruses are going to rely on surface interactions between the viral proteins and host cell receptors. And these receptors are not made for viruses. These receptors are normal cellular proteins or glycoproteins, but viruses have kind of evolved to exploit them. Once they're attached, the virus is going to gain entry, either as a whole particle again, or just by delivering the genome into the cell. And the type of genome, whether it's DNA, RNA, single-stranded or double-stranded, is going to then determine the synthesis strategy. But the early steps, attachment and entry are really critical in order for infection to begin. This attachment step is like the virus kind of looking at the host cell wall, except it has what it needs to kind of get in. So viral attachment is going to occur when specific spikes or those surface proteins on the virus recognize and bind to receptors on the host cell membrane. This receptor specificity is going to determine which tissues or species the virus can actually infect. So the example I used earlier was human immunodeficiency virus, or HIV. This is going to be able to bind CD4 receptors and co-receptors on T helper cells. If we're talking about SARS-CoV-2, this is going to use spike proteins, and it's going to bind to a different receptor, the ACE2 receptor in the respiratory and the intestinal tracts. So if the right receptor isn't there, the virus simply can't attach, which is why host range matters so much. It is also going to explain why some viruses can cross species. So if they mutate just enough to recognize a new receptor, then they can infect a new host. So once the virus attaches, the next step is getting inside of the host cell. So animal viruses can do this in 3 ways. The first way, is direct penetration. So this is seen mostly in naked viruses, those ones without the envelope. The viral genome is injected directly into the host cell cytoplasm, and that capsid is going to stay outside. The second one is membrane fusion. This is common in envelope viruses. So the viral envelope is actually going to fuse with the host membrane, and then the whole capsid is going to enter into the cytoplasm. After that, the capsid is going to disassemble, and then it's going to cause a release of the viral genome. So two that use this strategy are measles and HIV. The third type is endocytosis. So here the cell is essentially kind of swallowing the virus into a vesicle. Once it's inside, the virus is going to escape from the vesicle, shed its coating, a process called uncoating, and then begin replication. And this is common for both naked and envelope viruses. So things like influenza A and adenovirus. After it enters into the host cell, the uncoating step is going to occur. And this is where it's going to go in and remove the capsid and all of those capsomeres or proteins and expose the viral genome. So depending on the virus, uncoating can happen at the plasma membrane, it can happen in the cytoplasm, or it can happen after it's been moved to the nucleus. So this is really a crucial step because the viral genome can't be replicated or transcribed until it's free. As long as it's in the capsid, we can't get the host machine